It's a huge pleasure to be here. So I have many connections with Cambridge, and it's always a pleasure to come and speak here. Excuse me if I'm just a little bit frazzled because I got up at 6:30 this morning to be here, and I left my laptop charger at home, and so we've just been into town to buy one and come out again. So it's money well spent, money well spent. So what I want to do is to talk about some brand new data. So you're the first people to hear a lot of this, so it has to come with a health warning that the referees haven't got their hands on it. But I thought you'd be interested. It's both of these things are relevant to the topic of neuroinflammation, particularly in psychosis. So neuroinflammation in psychosis, hot or not? And the two studies I want to talk about are fairly recently completed and largely unpublished. So the Spring study is a multi-center, multimodal imaging study, part of which involves imaging neuroinflammation in the brain. Is there or isn't there neuroinflammation in the brain? And the Benjamin study is a clinical trial looking to see if an anti-inflammatory antibiotic called minocycline benefits symptoms of schizophrenia by blocking neuroinflammation. So I want to begin just by giving you three pieces of evidence of why we're interested, what's led to, what's a stimulus to the interest in inflammation in psychosis. And I'll use this thing, okay? I hope you all know what that is. That's a Manhattan plot. It's about genetics. I don't want to spend any detail on it. And these are parts of the genome going from chromosome 1 all the way to the end. And these are bits of the genome that are more common in people with psychosis and schizophrenia than the general population. And the big hit that comes out of all of them is the major histocompatibility complex on chromosome 6, which is all about immune mechanisms and recognizing foreign antigens and so on. So that's led to a great interest in that possibility. But nobody knows, what genes are involved. There are many other genes. It's very hard to unpick it. And anyway, it only accounts for a relative risk of about 0.1%. So it's not a huge influence. It's just the biggest signal that we have so far. And then secondly, you've seen Golam showed you this meta-analysis, so I don't need to spend any time on it. So this is in people with psychosis, and there are many studies being carried out where you measure circulating cytokines, these inflammatory markers like C-reactive protein and IL-6 here at the top, and the red lines are the effect sizes across all the studies showing that in first episode psychosis in blue, acute psychosis in red, that many of these cytokines are increased in concentration, and when people get better, sometimes those changes reverse as well. And then another thing that made it really hot was this publication by a New York journalist who developed something that looked like schizophrenia. Everyone thought it was schizophrenia, but her symptoms were completely resistant to treatment. And it was only when it was discovered that she had anti-glutamate NMDA receptor antibodies and had an encephalitis and treated her with anti-inflammatory therapy that she got better. So a tiny number, maybe, of people with schizophrenia have an autoimmune disorder, and maybe sort of form fruits of that could be more prevalent. Okay, so neuroinflammation in the brain, a lot of debate about what that is and how you define it. For me, I think it should be the same as the rest of the body. So we want to see a cellular element to it and a molecular chemical element to it, a humoral element. And so we would want to see signs of microglial activation in the brain. So the first response to a bacterial invasion and so on is macrophages get active and gobble them up, and the CNS equivalent are the microglia. Astrocytes are also glial cells, but they really support neurons, and they get active in inflammation. So these things all do get active in people who've had strokes or in Alzheimer's disease, any Parkinson's. All of these signs of inflammation get active. And so the question is, does that happen to some degree in people with psychosis? And particularly the microglia start secreting cytokines, which draws in all sorts of other cells. And that's the innate system. And then we have the antibody system, if you like, the adaptive immunity system, where you'd expect to see white cells invading the brain and the production of antibodies. And of course, you'd expect to see some sort of sign of neurodegeneration and demyelination, and that's a big topic, but evidence that some degree of demyelination may be going on and altered connectivity in the brain. So just to show you a bit in a bit more detail, so this is a picture of microglia, and they come in two forms. So when they're resting, there's no threat, no invasion in the brain, then they are highly ramified. They've got branches, dendrites everywhere, and they're connecting with lots of cells and monitoring what's going on. If anything starts going wrong, any sign of damage, then they adopt this amoeboid form, start moving around to move in and attack whatever the problem is, and if things go wrong, perhaps altering neurons and their connections. So these are the sorts of weapons that microglia have. They gobble things up. They release these highly reactive chemical species that kill living things, release neurotransmitters as well, activate prostaglandin systems, release the cytokines, attract other cells in. They release proteinases that break down the blood-brain barrier so that things can cross it as part of the immune response, and they are part of the system that gets antibody systems going. So two things happen to microglia, or two points I want to make about activated microglia, is that they, when they're active, they express a protein called the translocator protein, and this is a protein that sits in mitochondria, and nobody quite knows what its real functions are. It's to do with cholesterol transport, all sorts of things that this transporter is implicated in. And when microglia get active, as in those diseases that I mentioned already, then the microglia express more of this stuff, and you can tag it and make it a positron-emitting version of it and use a PET camera to image microglial activation. And these effects of activated microglia are blocked by this drug minocycline, an antibiotic, which is also anti-inflammatory, and it's used as part of the treatment of rheumatoid, for example, and other inflammatory disorders that don't involve infection. So a bit on the TSPO. So it really does go up in diseases involving inflammation, so that you can see it's up in this person with Alzheimer's disease. There are various ligands for the TSPO. This is PK1195, and I'm not going to go into the sort of the details of the other ones that are around. I'm just talking about ligands for TSPOs in this talk. And what's the evidence that minocycline actually does have an anti-inflammatory action in the brain? There's very little direct evidence. All the evidence comes from culturing microglia, perhaps from humans or in experimental animals, and doing things to animal brains. And minocycline is used as a tool to block microglial activation. But I'm just a little skeptical about how much it does that in the human situation, in the human brain. But here's some evidence. This is about it, actually. So this is in multi-system atrophy, and you can see one patient here followed on 2 occasions. It's getting worse. This condition is getting worse. And there's a very small study of minocycline in these people. And the point of this slide is to show that treatment with minocycline for 6 months attenuated the TSPO activation. And that's about as direct as we have at the moment. So minocycline is an antibiotic. It's neuroprotective. Those actions were discovered, and those were the first actions that really got me involved in this. We didn't really know so much about the rest of this 10 years ago. But we now know it's anti-inflammatory. That was known quite a while ago, but we more recently that it blocks microglial activation. It blocks programmed cell death in the brain, which is a fundamental principle of how the brain works. It interferes with glutamate neurotransmission in a good way. And even you can show some cognitive enhancing effects of single doses in about one study. And it scavenges up in a chemical way some of those reactive species that I was mentioning previously. So why was I interested in that? I guess this study really inspired me, but I've always wanted to do a study of neuroprotection in schizophrenia because of the common clinical experience that people present with severe psychosis. And there's usually been some sort of build-up. You can see it developing. In some cases, it's more dramatic than others. And if we could stop that, we could do something about stopping the onset or the most severe consequences of having psychosis. So these papers came out, and there was a lot of interest in progressive changes in grey matter of the brain at this time in the early 2000s. And so this is a study by Jeff Lieberman in the States, and it's a structural brain imaging study where people in their first episode, their first exposure to antipsychotics, treated at 1 year apart, and these are statistical parametric maps. So these hot areas are maps of statistical significance between the first and the follow-up. And you can see that both groups show evidence of loss of grey matter. There's less grey matter after 1 year in early psychosis. And this is a study that showed the same thing in the EU, multi-centre study, and even evidence that people with the at-risk mental state, before they convert, during the conversion process, lose grey matter. So that was the interest, using a neuroprotective agent to try and stop these changes. And so I was very interested in trying to use glutamate antagonists, for example, was this glutamate excitotoxicity. But for various reasons, and after several false starting trials, a couple of them that completely failed to recruit, we were so inexperienced and everything was wrong with them. But we eventually did a study of minocycline with my colleagues in Manchester. These are my two professorial colleagues, Imran Chowdhury and Nusrat Hussain, who are professors of global health. Pakistan has got about 300 psychiatrists for their colossal populations, and so they try and do stuff back home, and they have this Pakistan Institute for Learning and Living, and they developed an increasingly good clinical trials setup. So we got funding from the Stanley Foundation, treated 144 patients with minocycline for 1 year. It's amazing that they did it for that long. But we wanted to do it for a year to stop that grey matter change. We figured that you'd need a year to see progression. And you can see that negative symptoms were benefited. That's what we kind of expected, but just clinically significant, maybe 5 points on the PANS negative scale, positive and negative syndrome scales, and a rather selective effect. So on the strength of that, we have got funding from the MRC, the EME and the NIHR to carry out a multi-center study of Benamin, but this time to have some biomarkers in there to see if over the course of a year, it's the same design, minocycline would prevent the loss of grey matter, reduce circulating cytokines, which we measured at the beginning and all the way through, and improve cognitive performance. Eileen Joyce is here, she was responsible for that, and of course check that it worked. So here, the great and the good in UK schizophrenia research, who participated in this 6 academic centres with 10 UK trusts, 10 pharmacies, 10 R&D departments. Buy me a beer, I'll tell you the administrative burden in getting through this. It was horrific, but we got going and we succeeded. And this is a flow diagram just to show you the sorts of numbers that we did. So we randomized 200, having screened a very large number. So we have 2 groups of 100 entering the study, and at month 2, 8 weeks, a reasonable length of time for an acute antipsychotic trial. We had a good retention, and then all the way through month 6, month 9, month 12. So by month 12, we've lost a lot of people, but still there are 65 in the placebo group and 62 in the minocycline group. So it was a tribute to an amazing team that we had, and I think this is that's a picture of them. So, when we succeeded, we had a great night out, a great bunch of RAs, and they stayed with the study. Often in clinical trials they move and come and go and do clinical psychology or something, but they stayed with the study, and the manager, Emma and Kelly, were just fantastically keeping these going. So if these people had been average, we wouldn't have completed the study. They were outstandingly good. I was just a lucky so we recruited the right people. So it's good build-up, this, isn't it? So, and while we were doing it, the early studies with PET and TSPO came out. So should we be allowed to publish a preliminary publication where you've got 10 people in one group and 10 in the other, and this is the same thing. It's very small numbers, all treated with drugs, showing that patients had increased levels compared to controls of TSPO. So we're using an antimicroglial drug. All the evidence is suggesting microglia are up in early psychosis. Fantastic motivation. And indeed, this is perhaps a lesson. So we were doing this in Manchester as well, PK11195, and these were preliminary data, and never look at preliminary data. I'm sort of giving the game away a bit here, but this is what we found, a big increase in exactly the area of the brain that Jeff Lieberman's study showed changes in schizophrenia. So that, all more ammunition that we should see a beneficial effect, but that's what we saw. Absolutely zilch arini. And this is the negative scale, the PANS total, the positive scale, the Calgary depression scale, clinical global impression, and this shows the numbers retained, 80% retention at 2 months and about 60 odd after that. So absolutely nothing in it, no beneficial effect, either short term or long term. So that is a bit upsetting. And then this is the biomarkers, so we saw no effect on biomarkers either. So here are the cytokines. We measured them at the beginning, 6 months and 12 months. plasma IL-6 and CRP, and in the blue the placebo, in the red the minocycline treated. And you see there's no effect of minocycline, there's no change over time, minocycline doesn't affect cytokines. Have the rheumatologists measured cytokines during minocycline treatment of rheumatoid? No, there's not a single paper about what minocycline does to cytokines. I think it's only psychiatrists that measure cytokines as far as I can see, apart from CRP. No effect there. The grey matter volume, that was done here in Cambridge, the analysis of it. John Suckling, excellent guy. And you see in red on the left-hand side, this is this is grey matter volume in that Jeff Lieberman area. No change over 1 year. So we expected to see a decline that minocycline would prevent. And the same on the right-hand side. We measured cognition. There were no changes. We looked at the brain's response to doing a working memory task. No change over time, no deterioration, nothing for minocycline to work on. And so, of course, I've carved this up any number of different ways, as you can imagine. Try this group, try that group. Are we missing something? And so we looked for people who had the top half of CRP levels or IL-6, and maybe those guys would show a response. Of course, the corollary of that is when you've got no difference and you find a subgroup where you do have an effect, it will mean the other group are aggravated by the drug. So there's no way we're going to find a subgroup and then another group that wasn't aggravated by it, which it would have been interesting, but it's not there. Whether they were taking first or second generation antipsychotics could have been important. It doesn't seem to have been. Compliance is always a worry, but the patients were strangely honest in their compliance rating scales, and some of them saying, "No, I hate taking this stuff, I'm not taking it, I'm taking one a week," and others who love taking it, very involved in the study. And we were able, retrospectively, to get some blood levels as well. And if you just take the people who were very compliant with the medication, no difference between minocycline and placebo, either in the short term or the long term. I also checked individual centres. I do think that it's better if there's some way of centralising rating is terribly important, because even if you get high inter-rater agreement with the PAN scales, people are different in how they elicit symptoms. It doesn't mean that everyone is reliable. But so, no, there were no centres where there was an outstanding benefit of minocycline. So why was this trial negative when the previous study in Pakistan was positive? And one thought that somebody suggested to me just recently at the schizophrenia meeting in Florence is maybe in Pakistan, the greater poverty, the deprivation, the stress of living there, perhaps that's more of the sort of inflammatory route. We know that stress interferes, and obesity and starvation and so on activates inflammatory mechanisms, and perhaps that's why chronic exposure to that, not to mention childhood adversity, could have caused a positive effect. On the other hand, clinical trials are like that. Sometimes they work, sometimes they don't. Is minocycline a potent antimicroglial agent? The jury is out. Anyone who's got a neurodegenerative disease seems to be interested in minocycline, so it's been tried in stroke, Parkinson's disease. It doesn't work in motor neuron disease, and there's a negative study in Alzheimer's, I've just heard. So, and a good study. So, is it perhaps it's not a potent antimicroglial agent? We need to try more potent ones, possibly. That is going to happen. The people in the UK were a bit less severely ill than people in Pakistan, but maybe there's no microglial activation in schizophrenia for the drug to work on. And maybe microglial activation might be more important in other phases of the illness, very early on perhaps, or if people are very treatment resistant. But it didn't work. These people were, in the first 5 years, I should have said that, the illness it was meant to be early course schizophrenia. So nobody had more than 5 years of illness. So then the TSPO studies the negative ones began to come in. You always get that in the curve. And here are two negative studies that came out showing no difference between the patients that they studied and the healthy controls. And this is a slightly more complicated graph, but I won't, there's no point describing it in detail. There is no significant difference. If anything, a tendency in one way of looking at the data to even see reductions. So those are two negative studies. And then this study was published last year, or was it 2016? 2016 now, by Bloomfield from King's College, London. So it's great. We've got a sort of north-south ding-dong. It's always good to have a total disagreement with another group, especially when our southern competitors does our northern paranoia a great deal of good. And we're sure we're right. We must be right. So they found increases in people with schizophrenia. And all the studies I've shown you involve drug-treated patients. So that's always the problem with biological psychiatry, getting away from the effects of drugs. And the difference between this result and the by now largely negative studies is the way the data is analysed. So if you look at the way it's normally analysed with binding potential, volume of distribution, if anything, there's no difference, but if anything, it's reduced a bit in these patients. And they justify the method because other ways of doing it don't. There's a vascular compartment where there are microglia that might be important in what the result that you get. So they model it in a different way, and they also refer it to the whole brain instead of measuring it area by area. So the jury is out, but I think most people think that it's at least not increased in people with schizophrenia. And given what I showed you with Alzheimer's disease, we don't see anything like that at all, not even a trend. In our data. What am I talking? I haven't told you the data yet. Here it is. So I beg your pardon. So I showed you the premature increase that kept us motivated. This is a complete study that we did, and the study I'm going to tell you about is the Spring study, where we've done it again. So the key point is that the increases that I showed you during the course of the study were in drug-treated people. So Peter Talbot, who ran this study with Sophie Holmes, a PhD student, selected patients in their first episode, drug-free as possible, and so they have a group of drug-free people, and there you see there's no difference in the drug-free people. And another group, he selected people in their first episode who were treated with risperidone, and there you see the increase. It looks like risperidone is responsible for the increase that we got excited about beforehand. And this is the first study with drug-free patients. And that's the way the literature looks. So this is we've done this all again in this multi-center imaging study called Spring between Cardiff, Manchester, and Nottingham University. We're measuring MRS, MEG, and PK11195. And essentially we've replicated the negative findings. So we had two groups of people, young patients, recent onset, and a group of younger controls, and then people who'd had the illness for more than 10 years, and a group of older controls. And you can see straight away that there's clearly no increase in PK119 binding between the patients. There's clearly an effect of aging, but that is not modified by the diagnosis of schizophrenia. There's definitely a drug effect in this. I'm not sure if this is the next slide. I'm experimenting. This is a new presentation that I'm trying out on you guys. Sorry if it's a bit rough around the edges. We find that the drug-free people, 6 of the recent onset patients were antipsychotic free, and they had lower PK11195 binding than the age-matched controls. It's pretty clear in the literature now. I don't have time to go through it, but maybe sort of 3-4 studies, it's either not increased or decreased in drug-free patients, decreased in PK11195. What is the implication of that? So we've looked at whether it correlates with clinical features, and there is this finding that we have that it does go with negative symptoms. So even though there's no difference overall, and particularly in the older group, but across the board actually, the young and the old, I'm showing you the older group here. If you look at the PANSS negative scale, obviously just a schizophrenia group, the greater the inflammation as measured by PK, the more the negative symptoms. But overall, there's no difference between their controls. It must relate to something else in the controls, because clearly they don't have, at least I don't think they have negative symptoms. So and this replicates the first finding that I showed you. So the first finding showed a drug effect, this finding shows a drug effect, and also that's the study with everybody in, the antipsychotic free and the medicated, there's a correlation between the two, even though the groups don't differ. So there are three things, I think. No difference in 2 studies now from Manchester, and other studies are backing it up. suggestion of a decrease in people who are drug-free and a relationship with negative symptoms in both of the studies. So, to conclude, I hope I've not gone too quickly, neuroinflammation in psychosis, hot or not? Well, activation of microglia, the CNS resident macrophages. Minocycline doesn't do anything. It's supposed to be an antimicroglial agent, and no increase in TSPO binding to activated microglia, and probably decreased in drug-free people. And just to spend a moment thinking about it, people are beginning to think about what it might mean. Maybe the microglia are dysfunctional in not expressing even a normal amount of TPSO, and so that they have impaired immune surveillance, and that could give rise to some kind of immune mechanism for psychosis. But I like the idea that it's 75% inherited. These genes are all to do, or many of them, to do with neurodevelopment, and what we're looking at with circulating cytokines and the reduced TPSO is perhaps a failure to be able to prune properly, because the microglia, that's their job in health, is to prune back connections. So maybe the development of the brain could be affected that way. And that's for adaptive immunity. There is a trial, natalizumab, in progress, which works in multiple sclerosis, and that would be a real killer finding if that's negative. If it's positive, even better, but we'll have to wait and see. So, hot or not, I think the answer is lukewarm, but it's extremely interesting. It's opened up a whole new novel thing. What's that low TPSO that's disease-related, nothing to do with drugs? What is that about? Thank you very much.